Let's understand mitosis and meiosis. Mitosis is a fundamental process in cell biology, and it is a type of cell division that occurs in eukaryotic cells. It is responsible for the growth, repair, and maintenance of multicellular organisms. Here are the key points about mitosis. 1. Cell division. Mitosis is a form of cell division where a single eukaryotic cell divides into two genetically identical daughter cells. This process ensures that each daughter cell receives an identical set of chromosomes to the parent cell. 2. Purpose. The primary purpose of mitosis is to enable growth and tissue repair in multicellular organisms. It is responsible for producing new cells to replace damaged or old ones. 3. Stages of mitosis. Mitosis consists of several distinct stages, including interphase. The cell prepares for division during this stage, which includes the replication of DNA. Prophase. Chromosomes condense and become visible. The nuclear envelope begins to break down. Metaphase. Chromosomes align along the cell's equatorial plane, the metaphase plate. Anaphase. Sister chromatids are pulled apart and moved to opposite poles of the cell. Telophase. New nuclear envelopes form around the separated chromosomes, creating two distinct nuclei. Cytokinesis. The cell physically divides into two daughter cells. In animal cells, this often involves the formation of a cleavage furrow, while plant cells build a cell plate. 4. Chromosome number. In mitosis, the chromosome number in the daughter cells is the same as the parent cell. This ensures that the genetic information remains constant. 5. Genetic diversity. Unlike meiosis, another type of cell division, mitosis does not contribute to genetic diversity because the daughter cells are genetically identical to the parent cell. 6. Role in multicellular organisms. Mitosis is essential for the growth and development of multicellular organisms. It replaces damaged or old cells and maintains the overall structure and function of tissues and organs. 7. Regulation. Mitosis is tightly regulated by checkpoints within the cell cycle to ensure accurate DNA replication and cell division. Any errors or problems in these checkpoints can lead to issues such as cancer. 8. Mitosis versus meiosis. While mitosis results in two identical daughter cells with the same chromosome number as the parent cell, meiosis is a specialized type of cell division that reduces the chromosome number by half, leading to the formation of gametes, sperm and egg cells, in sexually reproducing organisms. In summary, mitosis is a crucial cellular process that allows organisms to grow, repair damaged tissues, and maintain their overall structure. It ensures that the genetic information is faithfully passed from one generation of cells to the next. Meiosis is a specialized form of cell division that occurs in sexually reproducing organisms. It is responsible for the formation of gametes, sperm and egg cells, with half the chromosome number of the parent cell. Here are the key points about meiosis. 1. Cell division type. Meiosis is a type of cell division distinct from mitosis, which is involved in growth and tissue repair. Its primary purpose is to produce haploid cells, gametes, from diploid cells, somatic cells. 2. Chromosome number. Unlike mitosis, which results in daughter cells with the same chromosome number as the parent cell, meiosis reduces the chromosome number by half. This reduction is essential for maintaining the chromosome number from one generation to the next during sexual reproduction. 3. Stages of meiosis. Meiosis consists of two sequential divisions, meiosis I and meiosis II. Each division has specific stages. Meiosis I, reduction division. 1. Prophase I, chromosomes condense, homologous chromosomes pair up, synapsis, and genetic recombination, crossing over, occurs. 2. Metaphase I, paired homologous chromosomes align along the cell's equatorial plane. 3. Anaphase I, homologous chromosomes are pulled apart and moved to opposite poles of the cell. 4. Telophase I, two haploid daughter cells are formed, each with a mixture of maternal and paternal chromosomes. Cytokinesis occurs, resulting in two cells. Meiosis II, equational division. 5. Prophase II, chromosomes recondense in the haploid daughter cells. 6. Metaphase II, individual chromosomes align along the equatorial plane in each cell. 7. Anaphase II, sister chromatids are separated and moved to opposite poles of the cells. 8. Telophase II, four haploid daughter cells are produced, each with a unique combination of genetic material. Cytokinesis occurs in each cell. 4. Genetic diversity. Meiosis contributes to genetic diversity because of the crossing over that occurs during prophase I. Crossing over involves the exchange of genetic material between homologous chromosomes, leading to genetic recombination. This process results in genetically unique gametes. 5. Role in sexual reproduction. Meiosis is crucial for sexual reproduction because it produces haploid gametes, sperm and egg cells, that can fuse during fertilization to form a diploid zygote. The zygote contains genetic material from both parents. 6. Regulation. Like mitosis, meiosis is tightly regulated by checkpoints within the cell cycle to ensure the accuracy of DNA replication and chromosome segregation. Errors in meiosis can lead to genetic disorders. 7. Differences from mitosis. Unlike mitosis, meiosis involves two rounds of division, meiosis I and meiosis II, resulting in four non-identical daughter cells. Mitosis, on the other hand, produces two identical daughter cells. Additionally, meiosis reduces the chromosome number, whereas mitosis maintains it. In summary, meiosis is a specialized form of cell division that reduces the chromosome number and plays a critical role in sexual reproduction, genetic diversity, and the formation of haploid gametes. It ensures that offspring inherit a unique combination of genetic material from their parents. The terms zygotene, pachydna, and leptotene are stages in the prophase I of meiosis, the first of two divisions involved in the formation of haploid gametes, sperm and egg cells. Prophase I is a complex and crucial phase during which genetic recombination, crossing over, occurs. Let's explore these stages. 1. Leptotene. Definition. Leptotene is the initial stage of prophase I. Characteristics. During leptotene, the chromatin in the cell nucleus starts to condense, and individual chromosomes become visible as long, thin threads. At this stage, each chromosome consists of two sister chromatids, which are exact copies of each other. 2. Zygotene. Definition. Zygotene follows leptotene and is the second stage of prophase I. Characteristics. In zygotene, Homologous chromosomes, chromosomes with the same genes but potentially different alleles, begin to pair up, a process known as synapsis. This pairing is facilitated by the formation of protein structures called synaptomal complexes between homologous chromosomes. During synapsis, segments of chromatids from one chromosome may align with corresponding segments of chromatids from the other chromosome, setting the stage for genetic recombination. 3. Pachydna. Definition. Pachydna is the third stage of prophase I, following zygoteny. Characteristics. Pachydna is a critical stage where crossing over, or genetic recombination, occurs. During crossing over, segments of chromatids from homologous chromosomes break and exchange places, resulting in a shuffling of genetic material. This process leads to genetic diversity among the resulting gametes. At the end of the Pachydna stage, the synaptomal complexes start to break down. After these stages, leptotin, zygoteny, and Pachydna, the cell proceeds through the remaining stages of prophase I, which include diplotin and diakinesis, before moving on to metaphase I, anaphase I, telophase I, and cytokinesis. These stages collectively result in the formation of two haploid daughter cells from each original diploid cell, each containing a unique combination of genetic material due to the events of prophase I, including crossing over during Pachydna. Diplotin and diakinesis are two subsequent stages of prophase I in meiosis, the first of two divisions involved in the formation of haploid gametes, sperm and egg cells. These stages follow the earlier stages of leptotin, zygoteny, and Pachydna. Let's explore these stages. 1. Diplotin. Definition. Diplotin is the fourth stage of prophase I, following Pachydna. Characteristics. During diplotin, several notable events occur. The synaptomal complexes, which held homologous chromosomes together during the preceding stages, start to break down. However, some chiasmata, cross-shaped structures, may remain at this point, marking the locations where genetic recombination, crossing over, took place during Pachydna. As the synaptomal complexes dissolve, the homologous chromosomes begin to separate slightly but remain connected at the chiasmata. Sister chromatids, which were closely associated throughout prophase I, may become more visible as individual structures. 2. Diakinesis. Definition. Diakinesis is the final stage of prophase I, following diplotin. Characteristics. In diakinesis, the following events occur. The condensation of chromosomes continues, making them more condensed and visible. Chiasmata, which represent points of genetic recombination between homologous chromosomes, are still visible and mark the locations where crossover events took place during Pachydna. The nuclear envelope may start to break down, preparing the cell for entry into metaphase I. After diakinesis, the cell progresses to the subsequent stages of meiosis, which include metaphase I, anaphase I, telophase I, and cytokinesis. These stages ultimately lead to the formation of two haploid daughter cells from each original diploid cell, each with a unique combination of genetic material due to the events of prophase I, including genetic recombination during Pachydna and the dissolution of synaptomal complexes during diplotin. Teacher. Vinod Arya. Contact number 63950467771.